mind, but
So what happened again?
that's all right. I don't know. Somebody must have hit my car in the parking lot. That's all right. We'll try to take care
Oh
of it.
man, look at that.
Yeah.
That's so bad, you know, when somebody
Mhm.
do something like that. It's yeah. I'm
Yeah.
sorry.
That's all right. That's just the most recent thing I just
Yeah.
found out about 10 minutes ago.
I probably need to forget about it right now and
Right,
focus.
right.
You have a very busy week in front of you, you know.
Oh,
So
yes, I do.
that car issue, yeah, will be okay if you just finish, your test and your exams, and then you will have more time,
Then I
to
can
figure
deal with
out
it.
what you can do. Yeah. Sorry. Yeah, it's bad. I know. And health. Are you healthy? Everything is good. Health wise, you good, and
Yeah,
everybody in your family is safe and healthy.
so far, yes.
Good. That's
I'm
good.
trying
That's
to
this
stay
is
that
this is the
way.
most important thing. Yeah. Okay. So as you know, this is the final week for this class, and you have two major exams coming. One of them for the lab, the final practical, and the final test. Okay. So for the lab practical, it is going to be exactly the same formatting like practical one. The lab that you need to study basically are the unknown enterobacteriaceae is going to be included in your test. Uh, Making yogurt is another one, ABI 20E test. Urinary tract, oral biofilm, bacteriophage, serology and immunology, DNA electrophoresis, and finally water analysis. Those are the labs that you are responsible for. Okay. Um, the That's going to be a tough lab, tough test. So I encourage everyone to study hard for that one. Okay. So before I go through this in more details, I like probably a few days ago, I sent you the grade distribution for your class, your course. And as you can see, you have the lab portion is 40%, and the lecture is approximately 60%. For the lab, you have two major lab practical. You already have done one done, and this is the second one is coming this week. It is going to be available for you starting tomorrow. So tomorrow at 8 o'clock in the morning, and you are going to have, hello? Hello?
Me?
Yeah, can you hear me?
Yes, yes, I can hear you.
Yeah, for some reason I don't know what's happening here. I
Uh,
can say.
I've got it on mute in case my
Yeah,
dogs
yeah,
bark or
that's
anything.
okay. So let me continue. So you have two lab. One of them already have you done. Have the another one which is coming this week. It will be start tomorrow. I will have it available for everyone from 8 o'clock in the morning, all day until the end of the day, Wednesday. Okay, and that is 100 point. Um, the Some of the lab assignments I didn't post the grade yet, but I really appreciate your patience with me. I'm working on them. So, I will have sometime, more time this week after I post the exams and the practical. So I will go ahead and start like grading those late assignments. The uh, the This is for the lab. For the lecture, 60% you have already done with 3 tests, and the final is going to be 150 points. That final is cumulative, but the way I will make it, I will try to make it easy for you. I'm going to include only one chapter from the previous chapter that were covered, and that's going to be chapter 5. So you have chapter 5. This is from the old or the previously um, you were tested in that one. But in addition to chapter 5, you have 5 more chapters, and those are the last chapters: 27, 28, 29, 30, and 32. So 27, 28, 29, 30, and 32. So total will be 6 chapters that you really need to focus on. The final worth 150 points, 150 points. It will be exactly same formatting like previous test. Multiple choice, true or false, maybe like few fill in the blank. It will have a little bit more essay, short essay. It will give you like some questions, and I need you to answer those like no more than like 4 or 5 lines at the most, okay, based on the question that you are going to see. This is for the final exam. The final exam is going to be available for you on Wednesday at 8 o'clock in the morning, and you will have 2 days to finish it. The end will be Thursday, August 6th, midnight. So you have the test available all this time. So I tried very much to have like an overlap between the final practical and the final test. So you can, if you start tomorrow, you finish your lab practical, you can take Wednesday off to study and prepare for the final, which is going to be Thursday. Whatever you like to do, you have the complete freedom to handle this. Okay, so do you have any question for me so far for this, for the grading policy and for the nature of the final practical and the final exam?
The only question I have about the grades, the extra credit that we have,
Yes.
I know one of those goes on the 3rd exam.
Yes,
So
I
do we
need
just
to, yeah, I will add
add
this.
that there?
Yes.
And then we have another one that was from the very beginning that said test your knowledge. Where do
Yes,
those points go?
that will go to the final.
For the final exam?
Yes.
Oh, great.
Yes.
Okay, thank you.
Yeah, that will, yeah. So for the lab, do you have any question for me for any of the lab or lab assignments or any lab that was not really clear for you? The lab is going to be kind of a tough one, the practical, and it will be like one question at a time. So there is no, you will not be able to track back. So you need to manage your time accordingly. Okay, because this is the policy for the lab, for the department, to have like each question, and you don't need to, there's no track back basically. Okay, what you need to know for the practical, especially for the all the biochemical testing that we had before, you need to know the nature of the media, used, if there is any indicator added, what is the name of the indicator, or if there is any reagent that was added after the experiment to interpret the result. You need to know the name of the reagents, and you need to be capable of differentiating between positive and negative results. Okay, so let me do this. Let me stop share. I don't know why. Let me do this. I will share with you one of those labs. So I will give you an example of how what you expect from in your practical. Yeah, just bear with me here. Okay, so let me share this with you here. So these are for the unknown Enterobacteriaceae lab. Okay, so what I meant by that, so for example, if I ask you, I give you like a tube, test tube like this. This is for the SIM test, and in this test, we were trying to characterize our unknown based on its ability to produce hydrogen sulfide and based on its ability to produce indole from tryptophan. So if I give you a tube like that and ask you if this is an SIM media, and is this a positive or negative for hydrogen sulfide production, for example. So based on the color, you will say, yeah, that is a positive for hydrogen sulfide production. Then you will see another question says, what kind of reagent did you use to detect hydrogen sulfide? Because hydrogen sulfide is colorless and you need to add something to detect it. So you need to tell me it is ferric ammonium sulfate, for example, okay? This kind of sort. If I same tube and I said, what about indole test? Is it positive or negative? So you can easily see the difference between a positive reaction and negative reaction. In positive reaction, if the bacterium is capable of producing indole, you are going to see this very nice pinkish reddish ring at the top of the broth. And in order to be able to see this, you need to add reagent. So I maybe ask you, what is the name of the reagent? You need to tell me that is Kovac, for example. So Kovac reagent was added to detect for indole. So this sort of question, okay? pH indicators are important. You have several pH indicators that were used. You need to know the names, the methyl red and the phenol red, for example. What is the difference between the two, and how they change color based on the acidity or alkalinity of the media. So you need to be able to differentiate between those two pH indicators. What else? You have another one like bromethymol blue. You need to be able to differentiate between bromethymol blue and brom chrysal purple. Two different pH indicator, two different color for two different tests. Okay, so also you need to tell you know how they look like if pH change in the media. So this kind of thing. So it will be lots of those kind of questions about the media used, the name of the media sometimes, what kind of pH indicator was used, how it looks, if it's positive or negative. I give you a tube like that, and then that tube was, say, this is like a Simon citrate media that was inoculated with bacterium X. And after incubation, this is how it looks like. So I will ask you if that bacteria was capable of utilizing citrate or no. Okay, you need to tell me that. Definitely based on the color. If the color is not changing to blue, so that is a negative results, meaning that the bacterium couldn't use or utilize the citrate as a sole carbon source in the media. I might ask you what kind of pH indicator, what is the name of the pH indicator? You need to tell me that is bromethymol blue. This is the name of the pH indicator. Okay. A test like that for amino acid decarboxylase. Okay. What is the pH indicator? Brom-chrysal purple. How it looks if it is positive or negative. So if you have a broth inoculated with bacterium and you add a specific amino acid, say like arginine or lysine, for example, and add the reagent, the pH indicator, and incubate. You look for the color. If the organism was capable of producing decarboxylase and removing the carboxylic group from the amino acid, the color is going to stay the same, no change. But if the organism was not capable of doing so, the media is going to turn to yellow. So remember always the purple is positive, the yellow is minus, negative. Also, it is important to know for this particular test, when we did this test, there was like an overlay of mineral oil after we did the inoculation. So you have the broth, you inoculate, and then you overlay with some mineral oil, and then you incubate. So why we put mineral oil? Because you need to create some sort of oxygen-free environment, because if there is oxygen, the organism is going to use the amino acid and go through straight to tricarboxylic acid cycle, so you will not be able to see any change, okay? So oxygen can be eliminated from the process by adding some mineral oil at the top of the broth. So this sort of, of question you are going to see. So any lab, you have any question for me for any lab or anything or any experiment that was not really clear and you really need to understand more about it?
I really struggled with, and I'm sorry, I don't have anything pulled up in front of me, but on the water analysis lab,
Yes. Yeah, let
the
me do this.
count,
Yeah, that is, let me see here what I have for the water analysis lab. It's always good when you have some question, because that will make this kind of like review more productive, because it will go straight to the point. So is why I encourage always every time to prepare some question for me so we can go through those directly. The water lab, that is an easy lab. Okay, let me stop sharing. Okay, and then I'm going to share the lab with you. Okay, so can you see my screen?
yes.
Okay, so which one, which question you,
Questions,
I mean, this is for water analysis. If you are working in a microbiology lab and you were asked to have like a water sample, and you need to test the water sample and making sure that there is no contamination with any fecal materials. This is the main idea. And why fecal materials in particular? Because there will be some pathogenic bacteria associated with it. So always there is an association between fecal, like coliform bacteria, okay, and those pathogenic bacteria. So you detect, if you detect in your water sample some like coliform, like E. coli or other coliform, then there is a big, huge possibility that that water sample was contaminated with fecal matter, and there might be some pathogenic organism there, okay? And the way you do this experiment, it is kind of like easy setting. So here you have the thing that you're using is, we call it Petri films, okay? Petri film, this is something similar to Petri plates, Petri dishes. It has the bacteria, the media already. It has the media, so it is saturated with the media, okay? It's kind of dry. So you take it, this is the media, and the media is kind of dehydrated. So when you add the water sample to it, that the water is going to rehydrate the media, and then you need to cover it to prevent evaporation. Okay, and when you do this, you are going to see the colony that is going to grow, if you have any, will appear like a kind of dots, okay? And in those colonies, there is some indicators, like pH indicator, like tetrazolium chloride. And that tetrazolium chloride is going to change color due to some respiratory activity going on, okay? So you will see, you can do two things. You can see the behavior, the color of the colony, and also you can see the behavior, how they like they produce this kind of bubble, different kind of bubbles, carbon dioxide, due to the fermentation process. So you can do counting and you can look at the bubble formation here. Here you have two different kind of Petri plates or Petri film, we call it Petri film here. And you can see those are dots here represent the different kind of bacteria. Okay, a coliform, in the presence of the indicator, usually they go this kind of nice red color in the presence of tetrazolium. E. coli is kind of different a little bit, it produces blue color. So if you have your water sample show a picture like that, so all those red dots represent coliform and all those blue dots represent E. coli. That will give you an indication that your water sample is not clean. It is kind of contamination, okay? And also you can directly count those spots and get a total count exactly same way like when you did when we do the colony forming units on Petri dishes. Exactly the same, using same kind of formula. If the plate very busy like that, so definitely you need to do some sort of dilution to be able to get an accurate number. Okay, so this is why if you look here, so this is your water sample. So you can take straight, like from the water sample, okay, and do dilution, 1 to 100 dilution, and then take from that 100 to 100 dilution, you take some aliquots, put it in the plate, in the Petri film, or you can take directly from the water sample, okay? So there are two different ways to do this, to take directly from the water sample without any dilution, or to make some sort of serial dilution to be able to do it. Okay, so which one is that you was confused about?
Well, number 1 and 2 was asking to do the calculations. And let's see, yeah, I said on the second aerobic one, there are 32 colonies. I didn't know if that was just a theoretical one we were looking at, or if one of those in the PowerPoint had 32 colonies and I was confused.
Yeah, okay.
I was just struggling. I couldn't really figure out what I was supposed to look at and then how to do it exactly.
For this one, okay, you say I give you a water sample. So you have two things you can do. First of all, you can get the total count for aerobic bacteria, aerobes, okay? Or you can get only the coliform, okay? So we are expecting that the aerobic bacteria will be too much. So you need to do dilution for the aerobic sample. You need to do dilution. So in the second aerobic perifilm, meaning that if you look here, okay, so this is your aerobic Petri film. Okay, this is your aerobic Petri film. So if you see in the aerobic Petri film, we have serial dilution because we are expecting to see lots of bacteria, right? So you have your water sample, you take 1 ml directly from the water sample and you make 1 to 100 dilution, okay? And you take from this 1 to 100, 1 ml, okay? This is the same here. What you did, you take 1 ml from this one, not from the water sample. So you have, this is number 1 treatment, number 2. So again, this is your water sample. You take 1 ml from your water sample that I provide you with. You make a serial dilution 1 to 100 here. And then take aliquot, 1 milli aliquot, and spread it over the Petri film to check for what you will see, okay? This is one. The second will be more dilution. So I will take from 1 to 100, 1 ml, from 1 to 100, again to 99 ml. So that will be 1 to 10,000 dilution right there. and then take from the 1 to 1,000, 1 to 10,000 dilution, 1 ml. So this is the second Petri film. So if I ask you the question here, it says, on the second aerobic Petri film, so this is your aerobic Petri film. This is your first Petri film, and this is your second. Okay, I will ask you this. Do you think, is there any, what you expect to see? Are you going to see more like bacterial colony on the first Petri film or the second Petri film?
I would expect the first to maybe be too numerous to count. And then the
Exactly.
second one is what you're going to look at.
Perfect. So why? Because this is less diluted. You take 1 to 100 and you take 1 ml from this. But here you take 1 ml from the 1 to 100 and diluted more. So I'm making 1 to 10,000 dilution here, and you take 1 ml. So here you are going to expect to see less count. So that was the question. So it gives you data. It says here on the second aerobic Petri film, okay, you have this number of colonies, 32. So what do you think? Can you do calculation based on this data? If I ask you, I gave you a Petri film, and I said you have 32 colonies. And I give you the amount plated, which is 1 ml, and I give you the dilution that was used. Can you do numbering? Can you do calculation?
I think so. If I look back at the formula, I
Yeah,
don't remember
the
what it was.
formula is so simple and so easy, and you need to remember it because you are going to see some questions for phage PFU also, phage forming unit, plaque forming unit. Same kind of same kind of formula, okay? And I'm still not very good in like using this, but the formula says the number, okay, of colonies divided by the dilution, total dilution.
I think it was in the PowerPoint somewhere too.
Yeah, times amount plated.
Okay.
Okay, this will be your formula here, okay? Let me do this. Can you see my writing?
Yes, I can.
Okay, so based on this, what is the number of colonies?
Was it 32?
Yes.
I don't remember.
Okay,
Okay.
yeah, 32. So you can remove this. And what is the total dilution from the second Petri film?
Well, would it be 1 over 10,000?
Yes, which is basically 10
Okay. Let's just
to
multiply it by
minus 4,
10,000.
right? What is the amount
Okay.
plated?
1 milliliter.
Yes. Okay. So that will be a very easy, simple math. It will be 32 times 10 to the 4. Okay. The mouse is acting weird. I need to throw it in the street.
Okay.
I can't believe it. Give me a hard time. This is the second mouse, you know, for this to happen. So if you look at this, that will be 3, 2, 1, 2, 3, 4 zeros, right?
Now, I would probably just write that as 3.2 times 10 to the 5th. Is that okay?
Exactly. Yeah, this is the best. This is what I need.
Okay.
Yeah, this is what I want. Then you put it like that, so you can see like that. Okay. And that will be the number here. So if you go back to this one, it says here on the coliform Petri film. So now look at the coliform. This is a coliform Petri film. Okay. So what happened in the coliform Petri film? We take directly 1 ml from the water specimen. Okay. We haven't done any kind of dilution. Why? Because we are expecting to see very low count of those. So always when we receive a water sample, we expect that there is no fecal matter contamination. So it is not really necessary to do any kind of dilution. So you take straight from the water sample, 1 ml. So here it says in that coliform you found 59 colonies. So how many total are in 1 ml based on this?
So you don't have any dilution number?
Mm-hmm.
So it's just 59?
Exactly. That's it.
Okay.
Well, we haven't done any dilution. Can you see any dilution here? You take 1 ml directly from water sample and you did count.
Okay, well, I totally missed those on my lab.
Yes,
Can
yeah.
you, will you look at maybe slide 8 for me?
Yes.
So those, the answer would be, assuming it was 1 milliliter, your answer would just be the 49 or the 87?
No, which one?
Or,
It's this question here.
right, like if, I was trying to go back and look at slide 8 and slide 6 also, because I thought in my mind I was looking at those. I was looking at that said the bacteria count was 143 on that first one. And so I was trying to use that number somehow along with the 32.
Yeah, no, there is no correlation.
Okay.
No correlation at all. The question here has no correlation with
Independent.
this. Yeah, question here it is straight. If you look at the question number 1, it is correlated to slide number
Right.
3. Yeah, there is no correlation. You don't need to correlate between those two. Okay.
But when it says, I'm curious on number 6, it says bacteria count is 143.
This one you
And
mean?
Yes. And that's different from colony count? Or
No,
does
it is,
it
yeah,
means the
because,
same thing?
yeah, this is basically, definitely, it is not one single bacterium here, okay? Because you have some media, right? This Petri film, it has dried media. And the media, as soon as you add the water sample, the media is going to rehydrate. And that allows the bacteria to grow. So they are going to grow, assuming that each one of those cells will give you like this dots, which is a kind of a colony, little colony. Okay. So we consider, based on the fact that each colony arises from one or a little bit more, maybe sometime more than one cells, fewer cells. If you look at the colony on the plate, this colony might came from only a single cell, or like few cells were together and they grew and multiply. So for the sake of argument, we correlate each colony with one cell. So when I say, okay, I give you a sample and ask you to tell me how many cells, bacterial cell. So you do colony and you correlate colony to the cell and you tell me based on the colony forming unit, you have this much cell in your sample, which is not 100% accurate because some colonies may arise from several cells, right? But again, that kind of, it is not going to be like something fatal. Like it will give you an estimation. Maybe it is not very, very accurate, but it is close at least. So when I say bacteria count or colony count, usually when we do colony, we do it on in Petri plates. But when you say bacterial count, because this is Petri film is supporting a less good or less growth factors than plates. Plates are more rich, more media, definitely more than Petri film. So this is why to differentiate between colony forming units and agar plates or like Petri dish plate and bacterial count on Petri film membrane. Because this is like a membrane. It's a membrane, okay, and there's 3M kind of a membrane, and it is saturated with it is the membrane itself is underneath the membrane there is a media. Okay, so you have the media here. You put the water sample for rehydration, and then you add the membrane attached to it. So the bacteria is going to grow on the media, and if it will be blotted against the membrane. So this is what you are going to see. You take the membrane out. Okay, it's like a blot. It's like a copy from the media. Okay, from this, and this is what you look for. So this is for the. You have another calculation which is for plaque forming unit, which you need also to worry about. You need to study this as well. So for this aerobic Petri film, you have any question for me here in this one?
No, I think I'm good now.
Oh,
Thank
okay,
you.
that's good. And let me do it. What about the plaque forming unit?
I couldn't find my mute button.
Yeah,
I think I'm okay.
with the plaque forming unit.
Honestly, don't remember. I know there was another lab. They've run together in my mind right now. I need to go
Is
through
it
and write them all out.
DNA electrophoresis?
I love that one.
Oh,
I
that's
had a
not-
question on that one. I'm not sure it's very important. I don't know if you want me to ask now or
yeah,
I
let me see.
emailed. Okay.
Yeah.
It was minor. It wasn't one that we had to answer, but in the lab it asked what would happen if you put 20 microliters in the well that holds 25 microliters. And I don't know if you read my email yet or not.
No,
I
that's
asked
okay.
about that.
Okay. So you said if you overload the
Well,
well.
no, it didn't ask if you overload it because it said it held 25 and what would happen if you put in 20.
Oh, you
I
mean
think unless I read it wrong.
it says the well can hold up to 25 microliter. So what happened if you put 20 microliter?
Yes, it said 20 instead of 2. It was somewhere.
That's a weird question, but it shouldn't be a problem, you know. Okay,
Okay, that's what I thought. That's what I thought.
yeah, for agarogel, basically, you have wells and you load your wells with the DNA. And remember, when you have a DNA sample, you mix your DNA sample with a tracking dye and with some like glycerol, some heavy thing that can like settle the sample, the DNA sample, not making it floating. Because if you don't have something like glycerol, for example, the sample will float and it will not settle inside at the base of the lane. And if this is the case, it will be floating and it will not enter the agarose. It will be in the buffer. Okay, so if you have a sample and you are not sure the concentration of your DNA and you need to load whatever you can, so you need to know the maximum capacity of your well. So if the maximum capacity is 25, you can load up to 25. But if you exceed the volume, that will float and that will be a problem. Okay, but if you load 20 microliters, that's perfectly fine. You know, it shouldn't be a problem.
I have an additional question on this lab. For questions 2 and 3, are we supposed to give like an approximate estimate based on the sizes that are given for lane 2, or because I didn't see any other type of way to get those
Yes,
sizes for lane 4?
exactly. Based on this,
Okay.
so you have okay. So you have your standard. Let me see here. Let me see what is this one here. Okay, so you have your lambda undigested with hand 3. So it's undigested, it is digested, and you know the size. This is lane 2 here. So you know the sizes, right? So when I ask you lane 4, which is this one, what is the size? So yeah, it's an estimation, basically.
Okay.
So you can say, oh, that this band here is this is 23 kilobase, and this is 9 kilobase. So now this one is somehow in between. It will be a problem if you that can be 10, that can be 20, right? Because you have a big, huge range, 23 and 9. So the best way when you do this kind of estimation is basically to measure the migration distance, okay, from the lane, okay, and get the size versus the distance that is migrated. And that will be a standard curve for you. And then you plot this based on, you measure the distance migration, and then you will be able, based on the information provided for the standard, to get a very good approximate of the size.
Okay.
So yeah, again, you have two variables. You have two things here. You can plot the size of your DNA versus the distance it migrates. Okay, this is for your known. Your unknown, you have only, you don't know the size, but you know the distance. So you can predict the size based on the migration distance. Okay, this is basically for this kind of question.
Okay, perfect.
Yeah, because here, what about here? If I ask you, you know, what about this one? Can you tell the or this one? Can you tell the size of these two?
Well, in comparison to lane 2, they'd both be smaller than
Exactly.
2.
So how much small? So the
Um,
last one here is 2 kilobase. And this is smaller than 2 kilobase, and definitely yes, because it migrates it a bit faster.
mhm.
But it could be 1.9, it could be 1.5. Okay, what about this one? It could be 1, it could be 0.5,
Mhm.
because you don't have anything here, so you
To measure.
can, yeah. But again, based on the migration, the distance, you will have a good idea about the sizes of those.
Okay.
Yeah. That's an interesting lab. Yeah, I wish we would be able to do this in real, like in reality, in the lab. It would be fun, but you are not lucky. Yeah. Do you have any question for this lab or any other labs?
I think I'm set. I think you answered the two I was most confused on was this
Yeah.
one and the water analysis. So
Okay.
thanks for the.
The plaque forming unit, do you have anything for the plaque forming unit, the phage, bacteriophage, the serology, immunology lab, the oral biofilm lab, urinary tract lab, any of those?
I don't have any off the top of my head, but was the correct answers to, I think, the oral biofilm, were those going to be posted at any point, or?
Let me see here. So oral biofilm. We do this. Yeah, bear with me. Okay, so let me share. Okay. So which question you have?
Well, I had missed one on the what I had sent in, so I'm not really sure which one I missed.
Okay, okay. I can tell you. Let me see here if I can grab your lab. So what is your name again? Julie?
No,
Joel?
this is Chloe.
Oh,
Yes.
Chloe. Okay. You and Julie have the same kind of voice to me. Okay, let me see Chloe here. Yeah. Let me see which one you missed. Yeah, I think it was question number 5. Question number 5, it says, why can the total colineforming unit be determined from the selective media used? Okay, remember, if I use a selective media to get, I will not be able to get the total number. Why? Because selective media, based on the name even, select. So that media is going to select some bacteria over the other. So it is not going to give me a true, real answer for how many, the total number I have. Okay, so for example, you have, you are using a media which select for, say, gram-positive bacteria, okay? So you are going to get only the number for gram-positive, and all gram-negative bacteria on the sample you are going to miss. Why? Because you use selective media. So you can't get the total count by using this kind of media. So you need to have a very rich media that will be capable of getting all different kind of microbial microbial cells in the sample.
Okay.
Yeah, that is what you missed. Yes. So
Okay.
I can't get a total count using selective media. I can't, because selective media will select only some over the other. Okay. And I give you an example again with, if I have a media select for gram-positive, so all gram-negative, I will not be able to get it from the sample. So I can't say this is a colineform number, the total number is this much, while I missed all gram-negative bacteria from the sample. Is what I'm trying to say.
Got it. Yes.
Yeah.
That makes sense.
Yeah. Any other question for this?
I think I'm good on the rest of them.
Good, great.
Yeah, I think I'm okay too.
Okay, so let's do your best, and the lab practical is going to be a tough one, so do your best to get it done.
And I
Yeah.
came a few minutes late. I know the setup for the practical, will it be the same, and or like how many questions was it?
Yeah, it will be the same exactly like practical one. Okay, the number of questions supposed to be around 50, 52, something like that. But in reality, what you are going to see when you are doing it, you will see the total number of questions around 30 something. Okay.
Okay.
Why 30 something? Because some question consists of more than one. So as I give you before, I give you like a sample, say this, what kind of media is this? And then what kind of bacteria? And then what kind of indicator? That will be in the same question. Okay. So they are made to be approximately 50 something.
Okay.
Okay. But you will see some question contain only one part, others will contain 3, basically. So you need to manage your time accordingly.
Okay.
And there is no track back. So you will not be able to, like to escape.
Yeah,
And
that's how
then,
it was, I believe.
yeah, because this is again the department policy for this practical.
Okay.
Yeah. Okay. Okay. Well, wish you the best of everything. Good luck for all of you, and always email me if you have anything that need to be clarified. I can record even like a my answer and post it for you. You don't need to to schedule for a meeting. So just email me your question, and I will go through it and explain it for you the best I can.
Awesome. Thank you.
Okay, thank you very much, guys. Good luck.
Thank you.
Thank you
Thank
very much.
you.
Thank you. Okay, bye-bye. Good night.
Bye-bye.
Bye-bye.